Welcome to edupediaworld.com. This is Somja Nair, your online biology tutor. In this video, we are going to discuss about mutations and genetic disorders. Mutations. Mutation is a phenomenon which results in alterations of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variations in DNA. One DNA helix runs continuously from one end to the other in each chromatid in a highly supercoiled form. Therefore, loss, that is deletions or gain, insertions or duplications of a segment of DNA results in alterations in chromosomes. Since genes are not to be located on the chromosomes, alterations in chromosomes result in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. Mutation also arises due to changes in single base pair of DNA. This is known as point mutation. A classical example of such a mutation is sickle cell anemia. Deletions and insertions of base pairs of DNA causes frameshift mutations. The mechanism of mutation is beyond the scope of this discussion at this level. Hence, we will talk about the topic later. However, there are many chemical and physical factors that induce mutations. These are referred to as mutagens. UV radiations can cause mutations in organism. It is a mutagen. Pedigree analysis. The idea that disorders are inherited has been prevailing in the human society since long. This was based on the heritability of certain characteristic features in families. After the rediscovery of Mendel's work, the practice of analyzing inheritance pattern of traits in human beings began. Since it is evident that control crosses that can be performed in a peat plant or some other organisms are not possible in case of human beings, study of family history about inheritance of a particular trait provides an alternative, such as an analysis of traits in several generations of family is called pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality or disease. These are some of the important standard symbols used in the pedigree analysis. Square for males, circle for females, diamond shape for sex unspecified, and affected individuals will be in red color, and crossing or mating will be represented by single line. Then parents and children will be connected with the lines. Genetic disorders. As you have studied in this chapter, each and every feature in any organism is controlled by one or the other gene located on the DNA present in chromosome. DNA is the carrier of genetic information. It is hence transmitted from one generation to other without any change or alteration. However, changes or alterations do take place occasionally. Such an alteration or change in the genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with inheritance of changed or altered genes or chromosomes. Broadly, genetic disorders may be grouped into two categories: Mendelian disorders and chromosomal disorders. What are Mendelian disorders? Mendelian disorders are mainly determined by alterations or mutations in the single gene. These disorders are transmitted to the offspring on the same lines as we have studied in the principles of inheritance. The pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. Most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome, as in the case of hemophilia. It is evident that these X-linked recessive traits show transmission from carrier female to male progeny. Autosomal dominant and recessive disorders. The first picture, that is for an autosomal dominant trait, that is myotonic dystrophy, and the second picture, the figure B, is for the autosomal recessive trait, that is sickle cell anemia. The first picture, that means in an autosomal dominant disease, if you inherited the abnormal gene from one parent, you can get the disease. Often, one of the parents may also have the disease. Dominant inheritance means an abnormal gene from one parent can cause disease, even though the matching gene from the other parent is normal. The abnormal gene dominates, so it will cause disease. That is happening in case of myotonic dystrophy. The second one, sickle cell anemia. It is an autosomal recessive disorder. That means two copies of an abnormal gene must be present in order for the disease or trait to develop. Genes come in pairs. One gene in each pair comes from the mother and the other gene comes from the father. Recessive inheritance means both genes in a pair must be abnormal to cause the disease. People with only one defective gene in the pair are called carriers. However, they can pass the abnormal gene to their children. Hemophilia. Hemophilia is a group of inherited blood disorders in which the blood does not clot properly. An affected individual may bleed spontaneously or for longer than a healthy person after injury or surgery. The blood coagulation mechanism is a process which transforms the blood from a liquid into solid form and involves several different clotting factors. The mechanism generates fibrin when it is activated, which together with platelet plug stops the bleeding. When the coagulation factors are missing or deficient, the blood does not clot properly and bleeding continues. Patients with hemophilia have a genetic defect which results in a deficiency in one of the blood clotting factors. This sex-linked recessive disease, which shows its transmission from unaffected carrier female to some of the male progeny, has been widely studied. The possibility of a female becoming a hemophilic is extremely rare because mother of such a female has to be at least carrier and the father should be hemophilic. That is unviable in later stage of life. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. There are two main types of hemophilia. Hemophilia A occurs due to factor 8 deficiency and hemophilia B due to factor 9 deficiency. Sickle cell anemia. This is an autosome linked recessive trait that can be transmitted from parents to the offsprings when both the partners are carriers for the gene or heterozygous in nature. The disease is controlled by a single pair of allele, HBA and HBS. Out of the three possible genotypes, only homozygous individual 4, HBS, that is HBS, show the diseased phenotype. Heterozygous HBA-HBS individuals appear apparently unaffected, but they are carriers of the disease and there is 50% probability of transmission of the mutant gene to the progeny. thus exhibiting sickle cell trait. The defect is caused by substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to the single base substitution at the 6th codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from bioconcave disc to elongated sickle-like structure. These are micrograph of red blood cells and amino acid composition of relevant portion of the beta chain of hemoglobin. Figure A, it is from the normal individual, and the figure B is from an individual with sickle cell anemia. Here you can see that in the normal individual, in the 6th position, glutamic is present. Glutamic acid is present, and in the 6th position, in the sickle cell anemic patients, valine is present. Phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine to tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of these in brain result in mental retardation. These are also excreted through urine because of its pure absorption by kidney. Chromosomal disorders. The chromosomal disorders are caused due to the absence or excess or abnormal arrangement of one or more chromosomes. Failure of segregation of chromatids during cell division cycle result in the gain or loss of a chromosome called aneuploidy. For example, Down syndrome result in the gain of extra copy of chromosome 21. Similarly, Turner syndrome result due to the loss of an X chromosome in human females. Failure of cytokines after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and his phenomenon is known as polyploidy. This condition is often seen in plants. The total number of chromosomes of a normal human being is 46, that is 23 pairs. Out of these, 22 pairs are autosomes and 1 pair of chromosomes are sex chromosomes. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one pair of chromosomes. These situations are known as trisomy or monosomy of a chromosome respectively. Such a situation leads to very serious consequences in the individual. Down syndrome, Turner syndrome, Klinefelter syndrome, and common example of chromosomal disorders. Down syndrome. The cause of this genetic disorder is the presence of an additional copy of chromosome number 21, that is trisomy of 21. This disorder was first described by Langdon Down in 1866. The affected individual is short, saturated with small round head, furrowed tongue and partially open mouth. Palm is broad with characteristic palm crease. Physical, psychomotor and mental development is retarded. This is a representative figure showing an individual inflicted with Down syndrome and the corresponding chromosomes of the individual. Here you can see in the position 21 you can see three additional copy. That is, one additional copy is present. Klinefelter syndrome. This genetic disorder is also caused due to the presence of an additional copy of X chromosome resulting into a karyotype of 47 and XXY. Such an individual has overall masculine development. However, the feminine development of breast, that is gynecomastia, is also expressed. Such individuals are sterile in nature. This is a tall stature with feminized character. Turner syndrome. Such a disorder is caused due to the absence of one of the X chromosome, that is 45 with X0. Such a female is sterile as ovaries are rudimentary besides other features including lack of other secondary sexual characters. In picture you can see a short stature and underdeveloped feminine character of a Turner syndrome patient. Thank you. In the next video we will be moving to the next chapter, the molecular basis of inheritance.